Okay, in this video, let's take a look at the process and reactions of glycogenolysis. So this is the opposite of glycogen synthesis. This is glycogen breakdown. Now this is an important reaction sequence because we now have entered the fasting state. And being in the fasting state, we have to now examine the effects of glucagon. Up to this point, we have remained in the fed state with insulin signaling. Now let's briefly go over the reactions required to release glucose from glycogen stores, because these reactions are the same in both skeletal muscle and liver. So this is the common point of these basic reactions, and then we'll talk about regulation, which differs between the two. So we have our glycogen molecule, and recall this is a combination of alpha 1,4 plus alpha 1,6 glycosidic linkages, and so it represents a very similar structure to starch, except that we have more branches, which is the alpha 1,6. So we have a very dense structure. The importance of glycogen is that we have lots of ends because of the branches in order to work on, so we can rapidly synthesize and break down glycogen. So the first step in this reaction is to add inorganic phosphate to this reaction to create glucose 1-phosphate. So this simple one-step reaction is going to release a glucose monomer from the glycogen polymer. The enzyme that does this is glycogen phosphorylase. Phosphorylase enzymes generally add inorganic phosphate to reactions for lysis. So instead of hydrolysis with water, we're adding a phosphate, so it's phosphorylase. Now the next step of the reaction is going to be mediated by phosphoglucomutase. Now recall, being a mutase, we are going to move a phosphate group within the molecule, so we are going to form glucose 6-phosphate. We are simply moving the phosphate from the number 1 carbon on glucose to the number 6 carbon. Now at this point, glucose 6-phosphate, this is the common sequence up to here for both skeletal muscle and the liver. At this point, the fate of glucose 6-phosphate differs. So in skeletal muscle, the purpose of glycogen breakdown is to supply glucose for glycolysis, because the need for this is based on the ATP needs of skeletal muscle. So the glucose 6-phosphate that is released from glycogen stores in skeletal muscle will be placed down the glycolytic pathway and ultimately generating ATP. However, in the liver, the purpose of glycogen stores in the liver is to restore and maintain fasting glucose levels in the blood. The glucose that is stored in the liver is not going to be used by the liver. In the fasting state in the liver will use an alternative source for energy, and those are fatty acids. So glucose in the liver, glucose 6-phosphate, will be converted to glucose. And the enzyme that does this is glucose 6-phosphatase. This enzyme cleaves the number 6-phosphate, releasing inorganic phosphate, generating glucose. The glucose that is now generated inside the hepatocyte of the liver can exit the liver using the glucose transporter 2, GLUT2 transporter. Recall that this transporter is on the surface of hepatocytes at all times. It is not moved from intracellular stores. It is not insulin or glucagon responsive. It's on the surface at all times and allows the passage of glucose into the blood. And this is by the mechanism, by the liver maintaining plasma glucose levels. Now recall that GLUT2, is a low affinity transporter of glucose. In the fed state, glucose enters the liver by GLUT2 because we have high concentrations of glucose in the blood. In the fasting state, we have high concentrations of glucose inside the hepatocyte. This high concentration will drive the glucose out of the cell and into the blood. Now, the most important part of regulation is going to be looking at the activity, the mechanism of these regulations, and then also comparing them between muscle and liver. So the enzyme that we are going to be considering in regulation of glycogen release is glycogen phosphorylase. Now in muscle, the regulation of glycogen release, glycogen breakdown, is controlled by energy and need. So the first level of regulation is AMP. As muscles start to contract, they are going to hydrolyze ATP into ADP, and then we can generate some AMP as our signal. This indicates a declining energy charge in the cell. When this AMP concentration begins to rise, it will bind to the glycogen phosphorylase, being an allosteric activator. This will essentially turn on the glycogen phosphorylase to release glucose from the stores to raise ATP levels. Now also recall that glycolysis is also being regulated because AMP is a positive regulator of PFK1. So the combined effect of AMP will stimulate the activation of glycolysis and also activate the phosphorylase to release glucose from the stores to place down the glycolytic pathway. Now this happens when we just generate a little bit of AMP, so we have a low activity in the muscles. What happens now when we turn it up a notch and we ask the muscles to start really contracting? Well, recall that in order to contract, muscle cells must receive a neural innervation. This innervation will trigger the release of intracellular calcium stores, which then go on to perform the contraction necessary for muscle, along with ATP hydrolysis. So the intracellular rise in calcium will activate an enzyme called glycogen phosphorylase kinase. And this works by direct binding of the calcium to the phosphorylase kinase enzyme. The kinase enzyme has a calmodulin binding domain, which will bind calcium and activate kinase. Now this kinase is going to do two functions. The first is it's going to phosphorylate the glycogen phosphorylase once it's activated, being a kinase, and this will activate the glycogen phosphorylase enzyme through phosphorylation to increase glucose release. The second activity of this kinase is to phosphorylate the enzyme glycogen synthase. Recall this is the enzyme that synthesizes glycogen from glucose monomers from UDP glucose. Phosphorylation of this enzyme will inactivate the glycogen synthase. So up to this point, we've seen that AMP, just from mild muscle activity, will increase the activity of phosphorylase by direct allosteric activation. But now when we turn up the muscle activity by increasing calcium for contraction, we also turn up the need for greater energy. So in this way, we can turn on a kinase that will further activate the glycogen phosphorylase to release more glucose for glycolysis, ATP, and the increased energy demands. The other factor of the kinase that we want to consider is that we are going to shut off the glycogen synthase. This will prevent what is called a futile loop. By inhibiting the synthase enzyme, once we release the glucose from the stores, this prevents this reaction from going backwards and resynthesizing glycogen in the fasting state. So we turn on the phosphorylase, turn off the synthase. Now in muscle we have another level of regulation, and that is called epinephrine. Now recall, epinephrine is the fight or flight hormone, and this is the hormone coming from the adrenal gland that is responsible for really turning up our metabolism in case we need to fight or flight from a predator. So epinephrine will work on skeletal muscle by activating through its 7-membrane receptor, G-alpha S. The G-alpha S will in turn activate adenylate cyclase, which will generate our second messenger, cyclic AMP, which turns on, activates protein kinase A. Now we know that protein kinase A is also another kinase activity like glycogen phosphorylase, and the function of protein kinase A, or pKa, is to phosphorylate the glycogen phosphorylase kinase. In this way, it will activate the kinase. Also, by activating the kinase, we can now activate further glycogen phosphorylase by phosphorylation and inactivate glycogen synthase further by phosphorylation. So this is a series of reactions that are essentially considered synergistic, so that AMP activates phosphorylase to release a little bit of glucose from glycogen stores when we need to recharge our muscle cells. Upon activity of the muscles with contraction, we want to increase the release of glucose further so that we supply full energy charge. And this is done by calcium binding directly to the kinase and activating it to activate the phosphorylase, inactivate synthase. And then when we get into fight or flight, we need maximal activity from our muscles. And therefore, we want to put the pedal to the metal and release glucose as much as possible and as rapidly possible from glycogen stores. So we are going to piggyback on these signalings from AMP and calcium by binding epinephrine to its receptor, which increases ultimately protein kinase A activity, which phosphorylates the glycogen phosphorylase kinase, activating it further from even the calcium. And then this kinase will activate glycogen phosphorylase even more and further inhibit the synthase to make sure that we have maximal glucose release. Now, the one hormone that we haven't talked about here is glucagon. Glucagon has no effect on the muscle. And this makes sense because skeletal muscle is not interested in the fasting state the hormone indicating the fasting state, because this indicates that there is a potential for energy need in the blood, and muscle is a closed system. It has the energy it needs in the form of glycogen, and when it releases glycogen into glucose, its job is to generate ATP for further muscle contraction. So muscle really has no interest in knowing that we are in a fasting state in the plasma. Now liver, on the other hand, liver does actually care. So the first response that liver will have will be to glucagon signaling. Recall, glucagon's receptor is also a 7-transmembrane receptor, which will activate G-alpha S of the heterotrimeric G proteins, which will in turn activate adenylate cyclase, increasing concentrations of cyclic AMP, and finally our cascade of activating cyclic AMP dependent protein kinase, which in this case is protein kinase A. Protein kinase A will in turn activate the glycogen phosphorylase kinase in the same manner that epinephrine activated the protein kinase in muscle. But this is now due to the response of glucagon, a fasting signal. So this phosphorylation event activates the kinase, and now the kinase will activate glycogen phosphorylase by a phosphorylation event, which will now convert glycogen into glucose 6-phosphate. In turn, this activated kinase will inhibit glycogen synthase, just as it did in muscle. Now, liver also has a response for fight or flight, because the job of liver is to ensure that there's plentiful glucose in the blood, in the plasma, and this is going to be released from glycogen stores. And so epinephrine will signal the liver to increase the glycogen phosphorylase activity. And it does so again by its epinephrine receptor, which ultimately leads to protein kinase A activation by the same sequence of signaling reactions. So the epinephrine will essentially activate further the G-alpha S, which will increase cyclic AMP further and potentiate the signal by protein kinase A. So these two signals will be more than additive on here to really ramp up the activity of the kinase to activate the phosphorylase to release glucose. Now, one final note is that in the liver, the glucose 6-phosphate will not be entering glycolysis. We have an enzyme that is called glucose 6-phosphate, phosphatase, T-A-S-E, glucose 6-phosphatase. This enzyme is responsible for cleaving the inorganic phosphate from the glucose 6-phosphate, releasing glucose monomers. And as we saw before, this can now enter the circulation through GLUT2 to maintain plasma glucose levels during a fasting state. So in summation, the reactions of glycogenolysis are rather simple. However, the regulation of this is critical because in skeletal muscle and the liver, we want a method to turn up the release of glucose. So when we just have small needs, such as in low energy, low activity in the muscle, or just with glucagon signaling to maintain blood glucose, we want to release glucose from glycogen stores at a modest rate. We don't want to go pedal to the metal. However, when we need extra glucose for ATP production, such as during neural-mediated contraction of muscle, epinephrine fight or flight reactions, also with the liver, then we want to maximize our glucose release from glycogen stores, and we're turning up the regulatory system.